Good afternoon, everyone. I welcome you all to our educational webinar series organized by Vijaya Diagnostic Center. Today, the session involves a brief discussion on yet another advanced methodology which evaluates the sperm DNA. Now, here the question is, why should the sperm analysis be performed at the molecular level? The answer is, since the sperm DNA also carries all of the instructions and information for the developing embryo, the genetic integrity of the sperm is a definite key consideration in infertility issues. To reveal more detailed information on today's topic, we have Dr. Asima on board. A brief introduction about the speaker. Dr. Asima is currently working as senior consultant pathologist at VDC since more than 3.5 years. She had finished her post-graduation from Kakatiya Medical College, Warangal, and obtained DNB certification from Apollo Clinicals, Kolkata. She had previously worked as consultant pathologist in major cities like Mumbai, Kolkata, etc., with 14 plus years of experience in the field. I request Dr. Asima to take over. Over to you, Dr. Asima.
Thank you so much, Dr. Pavani, and good evening, everyone. I am Dr. Asima here, and the topic of today, as told by ma'am, is sperm DNA fragmentation analysis. So let us start. Yes. So sperm DNA fragmentation analysis. Let us start with a brief description on the basis of the structure of sperm and sperm DNA. And also I'll be briefing about you about types of DNA damage, sperm DNA fragmentation, causes of sperm DNA fragmentation, the test and its types, then sperm chromatin dispersion test, and how this will help the infertile couples. The biology of the sperm, in short, as we all know, it has a head, a middle piece, and a tail. And the most important structure, the head, it consists of about 3 billion base pairs in the form of DNA compaction in the nucleus. Then going ahead, as we all know, the DNA function, the most important regions are the coding regions, and we all know that intact chromatin is critical for the production of a viable pregnancy. And DNA, it is a manual of instructions. So the main function of DNA is to provide intact the chromatin to the oocyte. And while doing so, there may be so many types of sperm DNA damage. So the most important for us here being the DNA fragmentation. Now, in the infertility field, there are some facts which I want to get in front of you. Male infertility is the commonest cause of infertility, yet we keep sidetracking it. It contributes to nearly 50% of the 15% couples of the reproductive age. And even now, the conventional semen analysis remains the routine test to diagnose the male infertility. But this cannot assess the sperm at the molecular level. So we need further testing. What is this sperm DNA fragmentation? It is the most frequent DNA anomaly present in the male gamete that has been associated to poor semen quality, low fertilization rates, impaired embryo quality, pre-implantation development, and reduced clinical outcomes in assisted reproduction procedures. It is the most common disturbance affecting the genetic material in the form of single or double-stranded breaks, which in turn may lead to male subfertility, IVF failure, and miscarriage. It can be caused by apoptosis, defects in chromatin remodeling during the process of spermiogenesis, and oxygen-induced radical DNA damage. The various causes of sperm DNA fragmentation. The most common cause is the oxidative stress, and this may be due to so many of the treatable or non-treatable causes. So, what are the causes? Infection, pyrexia, elevated testicular temperature, recreational drugs, smoking, alcohol, stress, diet, environmental and occupational pollutants, advanced age, varicocele, leukocytospermia, and sperm cytoplasmic droplets. And many of them, they are medical reasons which can be modified. So now why we are actually testing the sperm DNA? The answer to it is we have to, in the field of infertility, we may help to guide the couples with low damage to intrauterine insemination or IVF and guide those couple with high damage to intracytoplasmic sperm injection. Sperm DNA fragmentation also has a close inverse relationship with live birth rates. Latest studies were based on dividing couples into groups depending on the severity of their sperm damage, DNA damage. Those with fragmentation less than 25% had live births of 33% following IVF treatment. In contrast, couples with sperm DNA fragmentation more than 50%, they had much lower birth rates of 13% following the IVF treatment. So if we were to incorporate this new information into routine clinical care, we could direct these patients straight to the intracytoplasmic semen injection treatment, thus avoiding loss of valuable biological time, cost of failed cycles, and unsuccessful cycles of IVF treatment. Who are the people who may benefit from this test? The men who have explained infertility, those with arrested embryo development, with poor blastocyst development, multiple failed IVF and ICSI treatment, recurrent miscarriage, advanced age, varicocele, poor semen parameters, and exposure to harmful substances. What are the advantages of this test? It is a reliable test and it is beyond the routine seminal sample assessment of sperm DNA integrity, and it helps identify men who are at risk of failing to initiate a healthy pregnancy. It helps in the clinical diagnosis, management, and treatment of male infertility. It also has a prognostic value in assessing outcome of assisted conception treatment. Now, in the market, we can see different types of DNA fragmentation tests. The first of all, the sperm chromatin dispersion test, which we are doing at our place, and I'll be telling you in a little detail later on. The other tests are the common tests, and I'm sorry for the typing error here. It is basically an alkali denaturation electrophoresis test in which the sperms, they are treated with an alkali, and then we see that the, uh, the sperm DNA, it forms a comet, wherein the head of the comet will be the intact DNA and the tail will be of that of broken DNA, and this is due to the migration of the broken DNA in an alkaline medium towards the anode. So this is the principle of this test, which is being followed in some of the labs. It is a slightly, cumbersome procedure. Then coming to the tunnel test, this test is detecting DNA fragmentation by labeling the primers, the breakdown DNA fragments uh, uh, by probes, with the help of an enzyme known as as we all know, this enzyme, deoxynucleonyl transferase. This helps this substrate what we add to this attaching to the 3-hydroxy terminal. So after this substrate can be labeled using a fluorescent probe, and this fluorescent probe again can be detected using a fluorescent microscope or otherwise a flow cytometer also. Now, coming to the main test, what we do at the center, the sperm chromatin dispersion test. It is based on the principle that sperms with fragmented DNA They fail to produce the characteristic halo of dispersed DNA loops that is observed in the sperms with non-fragmented DNA following acid denaturation and removal of the nuclear proteins. It has three main steps: embedding of the spermatozoa in an agarose matrix. This we do to manipulate the cells. The second step will be the incubation in an acid unwinding solution, followed by a lysing agent. The acid solution acts as a DNA denaturant which dissolves the DNA double helix only in the presence of DNA damage, and the lysing solution is used to remove the nuclear proteins. So, by adding the denaturating solution, we see that this DNA loops, if they are intact, they come out in the form of a loop, and which is known as the halo. And the ones with the damage, they cannot form the dispersion halos, and they form a very minimal halo or there'll be no halo. After this, we have a step of washing, dehydration and increasing ethanol baths and staining, as we have to view this under a microscope. The halos of the spermatozoa in the samples can be classified. This is based on the sixth edition of the WHO into 5 categories: the large, the medium, the small, the without halo, and the ones without halo degraded. The ones with the halo width is in the large ones, the halo width is similar to or larger than the minor diameter of the core. Core is nothing but the nucleus of the sperm. Then halo size in between the large and the small halo will be the medium one, and the small one will have a halo similar to or smaller than 1/3 of the minor diameter of the core of the nucleus. And we also see that some of the sperms, they will present a core irregular, which is irregularly or weakly stained. This category is associated with severe damage affecting both DNA and the proteins. How to report or calculate the test results? Now, when we have an automated version, then which, like for example, we have SQA Vision, the results, they are automatically calculated and given to us. Then we can alternatively use the microscope also, wherein we have to calculate by the DFI percentage here will be given by the number of sperms with fragmented DNA divided by the number of sperms evaluated into 100. So the normal reference value is the normal ones will be with the fragmented DNA less than 15%, 15 to 30% good to fair enough, and then the abnormal ones are the ones with more than 30% fragmented DNA. What is the advantage of the STT method? It is very fast, simple, accurate, and highly reproducible. The total testing time is less than an hour. It is less costly. No complex instrumentation is needed, and we can also use the normal microscope. The results they are easily assessed by the technicians. Now, why test this? The couples will avoid the loss of valuable biological time, the cost of failed cycles, and the heartache of repeatedly unsuccessful cycles of IVF treatment. Unexplained couples, they will get a diagnosis and thus direct them to the best treatment for them. Happier, better informed couples, they will spread the word of the clinic's success. Clinics will have improved IVF success rates, a scientific rationale for using intracytoplasmic semen injection, sperm injection, and additional revenue from inappropriate male fertility testing. Their greater success rates will help them retain their patient bases, with couples settling even for their next children. At the outset, also I would like to comment on the test which we are doing at our center. We have nearly started this in June 2019. And when we started this, we generally had about 3 to 4 tests in a month. But as of now, we are getting nearly 20 to 25 in a month. And we have very trained personnel in the form of technicians and doctors also. And the very important thing here is meticulous slide preparation. So, uh, saying this, I want to thank you all for attending my session on DNA sperm fragmentation. Thank you so much.
Thank you, Dr. Asima, for enlightening us on the most interesting and a very relevant topic in today's world. The session is open for discussion. We may take a few questions from the attendees, if any. Uh, Dr. Asima, can you briefly describe the lab procedure for the sake of our laboratory colleagues, the
Yeah,
DNA
sure. Yeah, sure, ma'am.
fragmentation?
This is a technique in which we first see that whatever sperm DNA we have to check, we have to fit into a agarose gel so that we can manipulate the cells, whatever we have. So for this, we need a denaturating agent so that we unwind the DNA loops. For this, the important thing what we should have a denaturating agent, and we as we know it the DNA is coiled in between the proteins. We have proteins, histones, etc., which help in the base forming the base of the DNA. So these proteins have to be removed. So we are using a lysing agent for that, and of course for the fixation on the slide, we need a fixative agent. So first of all, like in brief, because I know my technicians are also attending this. So we take in the kit we have a gel, so that we have a background for the cells. This gel we have to melt in a series of temperatures, like 80 degrees and then 37 degrees. So for this we have a water bath. We melt the gel and then we add the semen into the gel. After that, we keep it for a few minutes in the incubator, and then we take out. And the important step in this is addition of the semen into the gel. After that, after mixing, we again keep this in the incubator for some time. And after removing that, we also have in the kit pre-coated slides. So, and we prepare the slide using this gel containing the semen. Afterwards, this slide is kept in the fridge for about 5 to 6 minutes, and then after that it is taken out. Sorry for that. After adding the semen and gel on the slide, we keep a cover slip. And the very important step here is the removal of the cover slip, because if we are lax in this, the total gel may come out and it will not serve the purpose. After removing the cover slip, we go to the next important steps like denaturation, lysing, and then followed by dehydration in graded alcohol reagents. And then finally, it is stained with right GMSA stain 1 and 2. And then it is ready now after drying to be viewed under the microscope.
Okay.
Was I
Any
able to
interferences? Any interferences during the analysis, test analysis?
Yeah. It is very important, first of all, that before doing this analysis, we have to know about the count of the sperm actually, because that depends on that we dilute the semen sample. It should be around 15 million. So there may be so many dilutions. So first of all, that is a thing. To prevent overlapping of the sperms, we have to be very careful about the dilution. Secondly, the temperatures. The temperatures, first temperature what we are putting that gel into is 80 degrees. So if we prolong that, there may be, burning of the sperms, etc. And we should see that we are adding the semen only when the semen, the gel, it comes to a temperature of 37 degrees. Before that, we cannot add. And And even in staining, we have to be a little careful. And as I said, while removing the cover slip also, as it we may, these are all the troubleshootings like what we all things can happen during the preparation of the slide.
Thank you very much, Dr. Asima. If there are no questions, we shall conclude on today's session. Now, the take home message is that testing on DNA fragmentation analysis earlier during the infertility evaluation process can ultimately save time, energy, and money, and also prevent the couple from undergoing more invasive procedures. We conclude today's session with a note of thanks and appreciation to the management of Vijaya Diagnostics for their support. I also immensely thank the attendees, IT, and the digital marketing team for the successful continuation of the series. Looking forward to our next successive meet. Thank you all once again. Thank you.